BB 2903 Lab Report 5 Fetal Pig Dissection Part 3 Circulatory, Respiratory, and Nervous Systems by Kellie, Chadwick, and Cirka.
Carbon dioxide is a product of cellular respiration. It passes from the cells into the bloodstream via the capillaries. Carbon dioxide is an acid that has the potential to poison the body if the human body had not developed a buffering system. A buffer is a substance that resists changes in pH when in contact with an acid or an alkali. The bicarbonate system is the buffer system that maintains the blood pH level of 7.4, creating a pH equilibrium. Carbonic acid is formed when the carbon dioxide and water mix. Carbonic acid then can dissociate into bicarbonate and hydrogen ions, creating a pH equilibrium. Refer to Figure 1. In this week's lab, we explore the buffering system of the blood by testing the amounts of carbon dioxide we exhale to the amounts of carbon dioxide in the room air. We also examine our own vital lung capacity.
In this lab, the goals were to examine the amount of carbon dioxide of the atmosphere to the amount of carbon dioxide exhaled. We also compare the amounts of carbon dioxide exhaled before and after exercise. To understand this, we also must understand how the buffering system of the blood works and to understand the structure of the respiratory system. Lastly, we explored our own vital lung capacities.
Because lime water turns cloudy when mixed with carbon dioxide, in the first part of the experiment, the amount of carbon dioxide in the room air was compared to the amount of carbon dioxide in the air we exhale. We predicted that the beaker containing the exhaled air would contain more carbon dioxide than the room air and therefore turn more cloudy. We expected the room air to either stay clear or turn less cloudy.
When exercising, the body produces more carbon dioxide than at rest. Respiration is the way the body removes carbon dioxide from the bloodstream. The medulla is responsible for controlling the rate of respiration. The chemoreceptors are stimulated by carbon dioxide. During exercise, one breathes faster to exhale larger amounts of carbon dioxide because the medulla monitors the pH level of the blood and regulates respiration.
The second part of the experiment compares the amount of CO2 in the room to the amount of carbon dioxide we exhale before and after exercise. Using phenolphthalein, this is possible because phenolphthalein, which was pink in the beginning of the experiment, turns clear when mixed with an acid, in this case carbon dioxide. A null hypothesis was formed that stated there is no significant difference in the amount of carbon dioxide in the three samples.
Vital capacity is the amount of air our lungs can exhale. The amount of air we can inhale and exhale is significant because the amount of air we can exhale represents the amount of air that our body can exchange from the environment to the cells of our body. The process of breathing is efficient because our 300 million alveoli make up a total surface area of about 807 square feet.
Refer to Figure 2. We average our vital lung capacities using a spirometer. We predicted we would fall within the second standard deviation of our class's average.
This lab was completed according to the protocol titled LAB5 Fetal Pig Dissection 3 Circulatory, Respiratory, and Nervous Systems, with the alteration to the procedure that the phenolphthalein was already prepared.
For the first part of the experiment, room air and exhaled air were bubbled into two separate beakers containing lime water for 1 minute. The lime water turned cloudy in the presence of carbon dioxide. As seen in Figure 3, the air exhaled into the lime water turned the sample cloudy, whereas the room air that was bubbled into lime water did not affect the cloudiness of the lime water. Our hypothesis was correct in believing that the exhaled air would turn the lime water more cloudy than the room air. This is because there is more carbon dioxide in the air that is exhaled than in the atmosphere.
The second part of the experiment, room air and exhaled air before and after exercise, was bubbled through 3 separate beakers of pale pink phenolphthalein. Phenolphthalein is a substance that turns clear when exposed to acid. The room air was bubbled into the beaker until it turned clear. The carbon dioxide is an acid. It will turn the phenolphthalein clear. However, the exhaled air before and after exercise was bubbled into the phenolphthalein for 30 seconds. With a transfer pipette, drops of ammonia, a base, were added to the phenolphthalein individually until the substance turned back to the original pale pink. An analysis of variance was performed on the section's data and then the entire class's data. A null hypothesis was formed which states that there is no real difference in the number of ammonia drops needed to turn the phenolphthalein back to the original state, meaning that there is no significant difference in the amount of carbon dioxide in the samples that were used. As seen in Figure 5, the p-value for the class is 1.8 times 10 to the negative fourth. Because the p-value is less than 0.05, this means that we reject the null hypothesis and that the room air before exercise exhaled air and the after exercise exhaled air contain significantly different amounts of carbon dioxide. As seen in Figure 6, the p-value is 1.8 times 10 to the 12th. Because this is also less than 0.05, we reject the null hypothesis and understand that the 3 air samples have a significant difference of carbon dioxide amounts.
The last section of this lab, we determined our vital lung capacities by blowing into a spirometer. We then compared our individual results to the class average and determined if our results fall within 2 standard deviations. As seen in Figure 7, both of us fall within the 2nd standard deviation of the class, and therefore we can be considered normal. However, as seen in Table 1, both of us are below the average vital lung capacity of our height. This can be explained by the fact that every individual is different. References.